started too far along, I wanted to kind of just remind us of the goals. And I think in the announcement that went out, we sort of talked about 3 things. We're going to try to understand models of investigator-initiated trials. I'm going to broaden that a little bit and talk about investigator-initiated research for a good portion of the talk. We'll talk a little bit about advantages, disadvantages, challenges in conducting this sort of research, and then share personal experiences. And along these lines, I think we all have personal experiences. And many of you have significant experience along these lines, investigator-initiated research, which is why we'd like to keep it a discussion. I don't think Rod or myself are here to say this is the way we do it. These are our experiences and what we've done along these lines. As I mentioned, I'm a clinician scientist. I do eye and vision research. I do investigator-initiated research. I also have federal NIH research as well, and do a mix of drug and device work that includes investigator-initiated research. Rob will probably talk a little bit about his background when I turn things over to him later in the discussion. Again, we welcome other people's input, experience, advice, counsel, what have you. So we just want to kind of get the discussion going. So the first thing I'd like to talk about is why I think about investigator-initiated research. I think there are several reasons. Number one, you could think it improves our general scientific knowledge on a topic, sort of an academic reason. From a company or industry standpoint, it can help support product strategy for the company, sort of a win-win scenario. I think that investigator-initiated research allows the academic to answer their own questions as opposed to industry-sponsored research in which you're handed a protocol and said, follow the protocol, et cetera, et cetera. So you're answering your own basic questions. This allows you to build your own and institutional profile, reputation, sort of rankings, and the like. The other thing I think to think about in investigator-initiated research is it allows you to diversify your lab's sponsorship portfolio. Sometimes they're tight with NIH or what have you. You've got another revenue source to keep your lab and folks going. You can obviously contribute to the improvement of patient health and well-being, conducting investigator-initiated research. And you may also be involved in supporting the new use or new indication for a specific drug or device along these lines. These are just some reasons. I'm sure there are others as well that you've experienced. Sort of on the flip side of that coin, I think it's important to kind of get out on the table. Industry partnerships are not without caveats, which we need to think through. There may be conflicts of interest that need to be considered along these lines, and important disclosure processes that we have. There may be pressures, both direct and perhaps indirect, relative to publishing or not publishing. For example, if there's a negative result and you feel as though that's an important contribution to the literature, but the industry sponsor may not, sort of that dynamic. And then there are sort of, I think, historical perceptions about conducting industry-based research that are important to think about. That is sort of that industry research undermines the sort of academic standards that we want to uphold, if you will. I think we're seeing a change in philosophy about that today, particularly with pay lines with federal sponsors that are as low as they are. We're also seeing, from the academic side, Provost's office start to look at faculty handbooks relative to conducting industry research and entrepreneurial initiatives and considering those more academic relative to scholarship today. I think most of these situations can be managed. We have institutional processes and systems in place to manage sort of these relationships, whether it be in our contracts, our reporting through our conflict of interest review boards, et cetera, et cetera. I think there are several things you need to think about and relative to investigator-initiated research. Number one, sort of what type you're talking about. You'll hear the terms IIR, IIS, IIT, and so I think it's important to use those correctly. So think about investigator-initiated research as being quite broad. When I think about investigator-initiated studies, I tend to think human studies, and then when I hear IIT, I think actual clinical trials, so interventional-based studies. Along those lines, in the IIR space or IIT space, you might have traditional IND, IDE sorts of studies and activity. You may have non-IND, IDE sorts of studies, drug, biologic, and device studies. You may have non-drug, non-biologic, non-device studies, perhaps chart review studies, natural history studies. And then also you'll have human or non-human or preclinical studies that are supported through these mechanisms as well. I think we need to think about sort of who's involved, our roles and titles, and we'll talk through this a little bit. What are our responsibilities, and in particular, what are each of the parties' responsibilities? Kind of challenges we have, what are the scope and criteria that people look for in IIR projects, and then how does the process happen, and what sort of review circumstances are placed around these sort of activities. It's important to remember that within the IIR space, and particularly when you're involving human subjects-based research, we have the typical standards of accountability that we need to maintain. That is, sometimes people think I'm working with industry and I'm just going to tell them I'm going to do something and they're going to write me a blank check and I can do whatever I want. That is not the case at all in this space. So we have federal guidelines, ICH guidelines, perhaps state law, institutional policy. The granting agency or contracting agency may have requirements. We have contractual agreements which spell out responsibility, timelines, milestones. Regulatory bodies are, of course, involved, whether it be the FDA or IRB, which includes approved protocols and investigator-based SOPs for this sort of activities. I just want to remind everybody of that. There are not a lot of data about what sort of activity are being being funded in the IIR space. I was able to find a survey that was reported in 2015 by the Association of Clinical Research Professionals that looked at this specifically. So they surveyed industry, and the respondents were primarily, they call them investigator-initiated sponsored research activities, so sort of a broad category, managers of these programs, and said, what is your company most likely funding? What buckets? And so you can see 89% of those respondents said we're funding products or studies of products with marketing authorization approval. In other words, I'm looking at a product, I want to understand something unique about it. I'm not necessarily trying to change the indication. I'm not getting an IND or IDE. I just want to understand something that I've noticed in clinic, if you will. Next up was outcomes research on patient outcomes only. Observational, 66% are funding observational studies. Products with unapproved product indications, investigational product indications, in vitro studies, perhaps preclinical studies, and outcomes research on pharmacoeconomics. So you can see the IIR space is somewhat broad in terms of what sponsors look at. I think some of us trialists are quick to think we're in the trial space only, but I just want to point this out that don't just think about trials in investigator-initiated work. Think more broadly than that.
Jason, can you take a question?
Yeah, discussion questions. I should have
As I look at those numbers, they seem to add up to a little bit more than 100%.
They do.
I'm wondering what the percentage actually means.
So check all that apply to you. So Novartis, fill out this survey, and do you fund this? Do you fund this? Do you fund this? Do you fund this? Yes or no? So that's why they add up to more than 100%. Okay. Other comments, thoughts, questions? I should have emphasized that more at the beginning. Okay, so I want to come back to the terms sponsor, investigator, funder, collaborator. And I think this is really important. A lot of us use these terms probably technically incorrectly. But it's important sort of we have the same baseline relative to using the terms properly. So to do this, you go to the FDA regs, section 3.12 of 21 CFR, and I've underlined, sponsor means a person who takes responsibility for and initiates the clinical investigation. So a lot of times we use the term sponsor to mean funder. That's not correct. So in the case of investigator-initiated work, and particularly that at an academic medical center, the sponsor is the academic medical center, university, the institution, and we are the investigator. If I was conducting this work outside of the private practice, I would be the sponsor-investigator. So oftentimes we will say industry-sponsored research. Industry-sponsored research technically is that which is drafted by, originated by industry. So just wanted to make sure we're kind of all thinking the same way along the lines of using these terms, and I probably will use it incorrect at some point, and you can call me out if I do. So sponsors' responsibilities, the institutional responsibilities for the 21 CFR are to select qualified investigators, sort of select, does the institution really select us? It's usually us coming to the institution with the project. Nonetheless, the institution is bound to have qualified investigators that perhaps may be trained and certified. I think we're seeing this initiative in the clinical trial space specifically. Munzer is here, and he runs the clinical investigator training program for trialists each year. So we have a robust training program at UAB. I think we're also seeing an initiative whereby we're looking probably in the future at needing to go through some level of certification relative to working in this space. It's probably not too far off in the distant horizon, individually and perhaps as an institution. Sponsors are also responsible to provide investigators resources they need to ensure proper monitoring of the investigation, that it's conducted, to ensure the study is conducted in accordance with the investigational plan, to maintain an effective IND or IDE if applicable, and to ensure that the regulatory bodies and all participants, including those in multi-site investigations, are promptly informed of significant new adverse events that happen in the course of the investigation. So I'm going to use the term collaborator here, and a collaborator in this instance means perhaps the funder, industry. So the collaborator responsibilities are much less than the sponsor responsibilities. So collaborator may provide funding and financial support of the project. Again, it doesn't mean they sponsor it. They may provide some protocol development assistance, typically a review through the review cycle. I'll talk about that in a little bit. But you are the originator of the protocol, or we are, the institution is in this space. They may provide provision of product, drug or device, and they also have some obligation to ensure regulatory compliance around the study. Again, sponsors, not the funder. So the IIR process varies a little bit company to company, but I think there's sort of an overarching model that that we see typically. I think one important point to emphasize, and this comes from OIG regs, the Office of the Inspector General, relative to compliance, the compliance program for pharma manufacturers, is that any remuneration from manufacturers provided to a purchaser that is expressly or impliedly related to the sale potentially implicates the anti-kickback statute. What that means is, the second sentence is, the second bullet point is the most important, to reduce risk on the pharma industry side, they in essence need to firewall marketing and sales from the grant review process. And I'm sure some of you have experienced this in the room, and typically that is very much the case. I'll talk a little bit about these review panels and what they look like in a little bit, but they need to firewall marketing and sales from their grant and contract review process. So looking at the review process, it's a little bit of a black box here, I think. But going back to the survey I mentioned before, surveyed about, I think it was about 100 different companies. 93% of the respondents of that survey said that we have a formal review process in the investigator-initiated research space. So they all say we've got a formal review process. The reviews range for those sponsors from 46 to 90 days. So once you put your application in, and you've got a little bit of time. The review panels on IIRs typically had a median of 5 reviewers, and these are the hats that the reviewers wear, so within the company. So you typically have a regulatory legal affairs person, typically a clinical person, typically a biostat or methodology person, your IIR, IT management person, and then typically safety, medical sort of affairs officer. So those are the people that will be looking at the applications. The respondents also said, I think that this is a really important point, greater than 50% of these respondents representing the industry indicated that IIRs have a greater than 50% chance of being funded in within those industries. And I think that that is an important opportunity for us to look at. I think that the investigator-initiated space is one that we're interested, UAB, speak institutionally, is interested in growing its activity in this space. Bob, you could probably speak for the School of Medicine agreement that we're interested in growing the investigator-initiated portfolio that we have. Maybe haven't looked at the numbers in a while, but I think we do between university executes maybe about 20 to 30 investigator-initiated trials per year. So that's about 10% of the activity we have that's funded by industry, and we'd like to see that grow.
How many companies were surveyed for this year?
I think it was like 100 or 110 or something like that.
So a pretty good cross-section.
I considered it a pretty good cross-section. Yeah, a mix of small, medium, large.
So in
So
my 40 years here, I've seen, you say it's greater than 50% chance of funding. It's highly variable. For some companies, it's less than 10%. Some companies, it's 80%. And I agree with you. They've tried to separate marketing from this process. Doesn't always work.
I think officially, that sort of knowing the right people, kind of talk about that a little bit, helps.
If I could just add an additional comment, that's 50%, greater than 50% chance of funding for proposals that are submitted. But there's a lot of discussion that goes on before submission that relates to the interest of the company in entertaining such a thing.
And
So-
if you don't do it, you have a less chance of getting it funded.
For sure. And the whole point there, the positive spin is to say the point is to help investigators keep from wasting time for proposals that are not of interest to industry. But what it does mean is the chance of funding as a percentage of submissions is a very different kind of number than investigator-initiated studies to the NIH, for example.
Yeah.
It's a very different space.
Good point. Good points, yeah. Okay, so I want to walk through a case example now, just to talk about process and expectations. And we've chosen Novartis here. And so we pulled their materials, just kind of walk through and talk about how it works. You can see this is Novartis's definition of investigator-initiated research. It can be, they point out, clinical or non-clinical. It's developed and sponsored by an independent investigator or academic sponsor. So they point that out, that we're the sponsor in these instances. Novartis may provide drug funding, drug, product, or both. So this is sort of the entry into their portal, if you will. I know you can't read this, but this is, I've blown them up a little bit. These are Novartis's requirements, sort of feasibility to conduct a clinical investigation. So I've blown them up. So they have investor qualifications. You can see current valid license to practice medicine, recent clinical research experience demonstrated within the last 3 years, good clinical practices training in the last 3 years, and a CV that basically shows that you can do this sort of work. There are study criteria. So these are broad, but the work you're proposing needs to have a legitimate research purpose with scientific merit that complements the Novartis-generated research. That's really, I think, a key, is that you want to sort of propose work that is a win-win. It helps the, I was going to say the sponsor. It helps industry understand their portfolio or their product, but it also answers a scientific question that you have, a clinical question that you have. They make a point to say that study criteria may help them better understand risk-benefit profiles and aligned again with Novartis compound scientific development strategy. Lastly, they point out that the investigator or the sponsor has to have the right infrastructure and resources to be able to conduct the study. I know that you can't read this, but I've blown things up. In essence, there are 3 phases of the Novartis process and most other processes, particularly for large pharma. The first phase is a letter of intent or a proof of concept that's submitted. Typically, these are submitted through the sponsor. They'll provide an email address, iit@novartis.com, in which you will write a 1-pager sort of talking about your concept idea. And in this case in particular, it's really, really important to have contacts at industry. So you've already sort of started the conversation. Perhaps it's a medical science liaison. or otherwise that you know to kind of sort of grease the skids, if you will, relative to this coming in. The letter is really important. It's basically a thumbs up or a thumbs down. At this point, if you are given a thumbs up, you're asked to submit a full protocol into their portal. And they have very mature and developed portals, most sponsors do. And there are two aspects to this submission, in particular for Novartis. If you follow on the left side, if you could read the print, it's the protocol. So we want to see exactly what you're proposing to do, your design, inclusion-exclusion criteria, your sample size, your endpoints, your timelines, et cetera, et cetera, et cetera. The right side is we want to see your budget, and we want to see it in detail. And the reason why we want to see it is because we're gonna examine it next to fair market standards. And so they will assess the budget and protocol at the same time. When you get to this stage, you would get perhaps a yes or no, or possibly a think about these things, and then come back up here and resubmit, depending on how your budget looked or how your protocol looked. Or if you're given a yes, at that point, then the industry will provide you a clinical trial agreement to go along with the protocol that you've developed. Renee Clements, who is our Associate Director for our industry group, is here for OSP, and she's in the back. And I think that officially, and I've talked to Melinda about this, officially OSP would like to be engaged in this process earlier. Okay? And I understand why, I've experienced why, because when you do include, when you include, for example, in your budget, some investigators might use a wrong F&A rate, or they might not include startup costs, things of that nature. I'm the first to admit that I don't sometimes go to OSP first. I will go through the process and sort of wait until it's approved, and then and I'm talking to Melinda Cotton, who is Associate Vice President and Director of OSP. She gets it. It's kind of a lot of work to put OSP through and not get it funded. So, but the official party line for OSP is we like to be engaged early and often. I have experience where I have submitted and used wrong numbers and kind of realized that after the fact, after it was approved, and the industry makes you go back up to starting and resubmit everything and then go through the process again. So there is legitimate reason to know what you're doing and work with professionals in OSP who are involved in the space. Then, once you are approved, you enter into the clinical trial agreement negotiation and also into the regulatory space with IRB, obviously for human-based studies. These are Novartis's expectations about budget and funding, and a few bullet points I've pulled out. I mentioned that they will look at your budget and do a fair market value assessment, and they have benchmarks. They know what all of our peers say, and so they have good numbers to sort of assess us against. Your budget must be justified. They can't include ordinary expenses that you would otherwise have for routine patient care, for example. And they have to be, your budget has to be milestone driven. And in particular, Novartis says specifically with these 3 milestones, we're going to give you 10, 10, 10. The other 70%, you need to figure out based on how your protocol looks. So they say at the execution of the CTA agreement and IRB approval, you get 10. Provision of the final third-party study report, you get 10%, and submission for publication and provision of publication of artists, you get 10%. Beyond that, then the 70% is for you to sort of work with whoever OSP or your department to figure out how you want those milestones to look. Relative to funding, when they will actually write the check, however, is when you have shown you have IRB approval and an executed clinical trial agreement. So they won't release any funds to you until that time. Now, I wanted to speak specifically internally about budgets. And so on our side, investigator side, it's really, really important to be all-encompassing in these budgets. So they must represent your full study costs. And just as a reminder, so we have startup fees, both institutional, and then your research group or department may have study startup fees. These may include things for the protocol development, which you've had to do relative to getting approval to move ahead in this space. Your regulatory filings, you charge for that. CFR generation, your case report form generation, for trainings, study initiation visits, whatever. Personnel, your procedures, your supplies, recruitment, monitoring, auditing, closeout visits, don't forget to charge for those, data storage, facilities, and your facilities and administration costs, so your indirect costs. Really important. My experience is, and happy to hear about others, is that the mature sponsors, I mean, these are line items in the portals that they sort of expect to see these values, and so they're just part of the portal application. Most of them are. And if they're not a line item, there are spaces where you can make them sort of a line item. So really important, though, that we get the budgets represent full steady costs.
Jason,
for internal use, we have developed our own document of the kinds of activities that are involved in the development and execution of clinical trials, trying to be complete, and then getting a rough estimate at what the typical costs involved with those activities are. As you look at that sheet, which we have internally and is available for distribution, how does that align with the line item activities that are in a portal like this? Is there pretty close alignment, or is that less clear?
Well, I think that there were sort of a lot of input on the development of the tool you're talking about, the internal document. And so we were able to really, I think, broadly cover what you might anticipate in an investigator-initiated trial relative to the internal budgeting document that we've prepared. I think there's great agreement here. I mean, it's a very comprehensive document that we created.
So would it be fair then to say, for those who are developing these budgets for submission to industry, there are some tools available to help them?
Absolutely.
In that regard?
Yeah, we can make them available I mean, they are available now, but we'll certainly promote those and make them part of the clinical trial initiatives we have.
Thank you.
Good point. Thanks for drawing that out. This is a table of responsibilities that Novartis expects. The 3 checks are Novartis responsibilities. The 27 checks are the sponsor's responsibilities. I know that you aren't able to read it, but I just want to point out that the onus is on us relative to responsibilities as a sponsor and as the investigator in this space in particular.
And we'll post all these slides on the website, so all of this will be archived for people to look at later.
Perfect.
So they can read all of the little tiny check marks.
Okay.
Cool.
I want to make a few points about the multi-site study initiative as it relates to the investigator-initiated space for just a moment. I mentioned that I wear that cap for the CCTS, and so I want to talk about 2 initiatives in particular. NCAPS, which funds our CCTS, is working on an initiative called the Trial Innovation Network. So think of it as a national network connected by the CTSA hubs, kind of a clinical trial superhighway. And you can see the roads between the hubs. So from a national federal standpoint, from an NCAT standpoint, we've created this network of hubs, and we have a robust working group, and I'll talk to this specifically, where we are working to both develop federal and industry trials and then push them out to the hubs for activity, for our involvement, if you will. So that's the national initiative. Locally then, so we are the hub, UAB, and we have a robust partner network, which we're calling SHARE. And so you can see our Southeast regional partners mapped out. We have a 14-member regional consortium that's sort of centered around the hub of some clinical partners and some non-clinical partners, in which we are developing this sort of translational network, such that as we or our network partners develop clinical trials, clinical studies in this space, we're able to go to whoever, Ochsner, Tulane, LSU. We work together. We have a monthly call the organization does where we look at studies, promote studies within the regional network. So this is a resource to us specifically in the space. Both the SHARE regional network and the Trial Innovation Network, the national network, looks at a variety of different studies. So think about, we can target different parts of the country for ethnic diversity, demographic variety, The network, particularly the Trial Innovation Network, looks at studies that are thousands of people. And so we're able to leverage the CTSA hubs along these lines and then their regional sort of networks. We're able to look at things in terms of rare disease. Source of funding is broad for these, so we would consider industry, federal, or foundation sources of funding. You can consider both of these networks for both trials in addition to observational studies, so perhaps prospective history studies, case control studies, if you will. So really, we're looking for all sorts of activity within these networks. The resources that we have available to us, UAB and partner community, are very broad. So we have single IRB support through the SMART IRB network. We have all agreed to and adopted sort of a common contracting method that includes both originating contract and I think subcontracts as well. So theory, we have agreed to common language, which in theory should improve and accelerate contracting times. We have protocol development assistance available to us both through the TIN and also locally through SHARE. Methodology, design, biostats, in particular through BIRD here on campus, and David Redden, I think, who I saw hovering around, and others. We have an ability to rapidly sort of locate PIs and resource identifications, particularly in our partner network. So for example, we had a trial come to us from Mansour through the Cancer Center looking at the effect of antiemetics and cancer. And in particular, we needed to have BMT units. And so we were able to go to our SHARE partners and say, are you interested in this study? And who has and who doesn't have BMT unit? And roll people in or out really quick along those lines. We also have the ability to leverage for recruitment, enrollment, and retention services, both traditional and electronic based. I think Jim Smino was here a little bit earlier and has done a lot in the electronic data warehouse I2B2 initiative. And we're in the process of pulling together nationally along these lines, in addition to regionally through the ACT network, to use these resources to be able to do rapid and efficient cohort identification. This is all free to you. The one thing that we don't do, either SHARE or TIN, are fund your studies. So you would still actually use your ICs and industry partners or whoever to fund your activity in the space. SHARE, our regional network, is a CCTS initiative again. We've got information here. Franny is the point of contact. She's your main person to go to. But a lot of us are involved. So Bob's involved, Jennifer's involved, I'm involved, others are involved. And again, we can help you with contracting or single IRB or training, et cetera, et cetera. The Trial Innovation Network, the national network, is an incredible resource. I think they've had, they both do consultations for studies, and then they will push out studies to the network. So we've had, we've interacted with them. About 20 studies, I think, have come to us, UAB opportunities. But then we're also able to go to them and say, we want to engage this 64 hub network, if not their regional as well. And so that happens through us, internally. You can't go to them directly. You have to work with us and the SHARE group to go to them. But it's a resource that's available to you, particularly on your very large studies along these lines. I think the key to success in using these resources for SHARE and TIN, I want to emphasize, Franny emphasizes early on. So if you get up to the point where you're already got an IRB in or you're contracting, that's sort of a little bit late. It doesn't mean we can't add sites or engage these networks per se, but the best time for you to approach us is early on when you're thinking about the multi-site study, you're thinking about potential partners. Contact us then, because that's the time that we can engage these resources early on and often. Okay, I want to sort of switch gears. Oh, is there sort of, I guess, pre-written?
So I was asking, is there pre-written language that you guys can help us with for grants if we want to have the innovation network and a grant proposal? Can you give us some?
Oh, you mean language about the research?
Yeah, so it's like if I want to say I wanted to use the innovation network or the SHARE,
absolutely.
There's like a paragraph you can send us that's already done.
Yeah, absolutely. Yeah, and the CCTS offers other resources as well, panels in particular for assistance with review, et cetera, et cetera. So the resources are really, really robust along these lines. So I want to share just a moment my personal experience that I have in this space and how I've made it work for me. And in particular, it sort of centers around the idea, which is really, really important. I think that the idea really needs to create a win-win scenario. So it needs to be a win for you, the investigator, and it needs to be a win for your partner, your industry partner. Sort of one-sided approaches where it's all about you, not as successful. I think it's really critical to be entrepreneurial in this space and really leverage your lab strengths and your unique abilities. And then also, it's really key for you to know the business that you're in, know sort of what the company's trying to do and understand, have a good contact and connection with your medical scientific liaisons, the MSLs, or folks in marketing or science, so that they kind of can help be your advocate internally on these initiatives. So I do eye research, and in particular, I do front of the eye research, so cornea and tears. And you can see the cornea is a structure, a clear avascular structure here that's about 550 microns thick. On the front of that is a tear layer that's about 4 microns thick in total thickness. And if you expand the tear layer, like in the upper category, there are 3 primary layers, if you will. We don't like to call them layers so much anymore. There's an outer lipid layer, which is about 40 nanometers thick. There is a sort of aqueous layer, which is sort of bound most posterior by mucins, membrane-bound mucins. And that sort of is about 3 point something, around 4 microns, let's say. So this is a really thin film that's exposed to the atmosphere. And you can see one of the effects of altered tear film or dry conditions can be sort of the picture of the eye base, which is you've got severe desiccation, drying, you get staining of the ocular surface, you get vascularization into the cornea, which is supposed to be avascular, and you can get scarring and loss of vision. So that's sort of the end result in severe dry eye or ocular surface conditions. Sort of says why the tears are really, really important. By the way, so 4 microns, anybody have any perspective? So a human hair, anybody know the diameter of a human hair? 10 microns multiplied by 10. So the human hair, a brown human hair, is about 100 microns. So this is a really, really thin film. And in particular, this lipid layer on the front, they said is 40 nanometers, really, really, really thin film. So what I've been able to do is fairly successfully is through my training and background, I purpose-built optical systems in order to image the tears in the eye. And these have to have ultra-high resolution. In fact, the most current system is this system here, which we are in the process of patenting, has the ability, it has an axial resolution of less than a micron, so super high resolution, at least in OCT terms. What we've done is couple the super high resolution OCT, optical coherence tomographer, to a microscope, which is able to have the resolution to examine the lipid on the front, so the 40 nanometer film. So we've coupled these 2 optical devices together and are able to look at sort of structure-function relationships. And these are, so this is a picture, this is like a topography picture from the microscope. So we have, I don't know, probably 10 nanometer resolution of microscopy. And that's a human cornea. You can see the cornea is about 550 in full thickness. The precorneal tear film's at the top of about 4 microns in thickness. So we can look at these two layers simultaneously. We've co-registered these systems. And so, I mean, for some reason the video wouldn't work here. So this is an example of what the tears look like. See this person will blink. You saw the lashes go down, come back up. Tear film floats back up. Actually, counter to gravity, there's really strong surface tension forces that pull the film up. And then you can see, as the eye sits here, in this particular case, a very stable film. You don't see much breakup, maybe a little bit here and here. No exposure to the ocular surface. So this is good. That's an example of good. Oop, didn't mean to do that. This is an example of perhaps not as good. So you see a lot of fringes. All of a sudden, you start to see breakup here. You start to see exposed surface. So that's not as good. And this is an example of really bad. Immediately after the blink, you see surface of the eye. There's no film covering it at all. So completely dry right out. So how is this opportunity, or how has this technology allowed me to sort of leverage the IIR space? I'm the only one in the world who can do these measures, particularly both simultaneously. And we have the highest resolution OCT that's been purpose-built to measure the front of the eye tear dynamics. Because of that, the eye industry says, we want to use that to understand how our drop, how our pharmaceutical, how our device, what's the impact, what happens to the tears, how do they settle, et cetera, et cetera. So we've really, really leveraged this strength to work with industry because we're the only ones that can do it. So we've been very fortunate along these lines, and that's sort of what's really worked for us in my lab in particular. So that's my story. And at this point, Rod, can you hear us?
I can. I've been on mute here for a while. So.
All right.
Can you guys hear me okay?
So I click here.
Not quite loud enough.
We can hear you fine.
That would be a first if people couldn't hear me. Ask any one of my family.
Doesn't want to blow up.
Yeah. So I'm going to go ahead and share my screen. All right. Can you guys see the screen okay?
Yes.
Yep.
Well, you know, the first 2 hours of my talk go by really quickly, and then after that, no, I want to thank Jason for doing an outstanding job. He did all the heavy lifting on this talk and discussion, and so I just really kind of wanted to point out a few personal experiences. And you know, first off, I did all my medical training there at UAB. Did med school residency and fellowship in oncology. Finished in 2007 after that short 11-year stint, and came down to LA, Lower Alabama, to University of South Alabama. And I found out after a few years working here, I did a little bit of everything, you know, clinical work, had my own basic and translational lab that focused on, you know, GYN malignancies, particularly starting off with cancer stem cell work, and that gravitated over time. But then also, you know, had an interest in clinical research and clinical trials. And I found out after a few years that if you complain and moan about something long enough, you might get put in charge of it. And so that was my experience. As the director at that time said, "Okay, big boy, why don't you see what you can do with the clinical trial mechanism here for cancer center?" So I've been the associate director of clinical research here since 2011. And so I wanted to hit on some of the failures, some of the successes. I'll talk about failures first. And I'm an optimistic guy, but kind of here we go. There's a lot of changes. As we know and understand, with any clinical trial mechanism and working with industry sponsors, there's a lot of change. One specific example, I had a company change. It really kind of was a failure. There was a new agent, and it was about in a pipeline in a small biotech company. And as many biotech companies do, they'll get to a certain point and they'll get bought out by other parent companies, and there's a lot of shifting and moving on. So this particular agent that I was working with this small biotech over a 4-year period, it had 3 changes during that time. It ended up being with Celgene, and I was young, naive, stubborn, all of the above. Kept pushing on, and finally when I got to Celgene, they just decided that this product didn't have enough efficacy, and the cancer line that I was looking at did not have enough cancer patients, so they cut the program. And that was a lesson, and Jason hit on this, is really kind of knowing that landscape that helps your proposals be better. Another failure, leadership change. There was a new hedgehog inhibitor that was in a pipeline at one of the top 3 cancer pharmaceutical companies. I was actually in the area of my research in cancer stem cells, looking at the hedgehog pathway, and I had preclinical data from in vitro and in vivo that was promising. Flew up to New Jersey to give a presentation of my work. Everyone was excited. The vice president of research and development said, "Yes, let's do this deal." Worked on a contract on further study, and within literally 2 weeks of that very promising meeting up there, He was fired. A new research and development person was hired, and they cut that drug and cut that program. So again, out of my control, but just knowing the landscape of how this kind of can change, especially dealing with industry and where their focus is, on the drugs, on, patients and improving healthcare, but it's also the bottom line that drives it as well. Competing approvals. This is something I think is interesting to me in the cancer world because it actually has changed such over time. When I was a fellow, I started working with a new low molecular weight heparinoid with a faculty mentor there at UAB. We had some preclinical data that looked promising for DVT prevention in perioperative setting for patients undergoing GYN cancer surgery. A Noxacan study came out, which is with a Lovenox showing a significant difference in reduction in postoperative DVTs, VTEs. And essentially, this program was cut from a competitor getting approval. I think it's interesting in the FDA's approach over the past few years to where that's really no longer the case. It used to be a first to market, and everyone else shifted to other areas. And there was a mad race to be first, and then second and third place, much like the Olympics. Most people don't remember. That's how pharmaceuticals consider that. But we've got right now, within the past 18 months, 3, FDA-indicated PARP inhibitors for ovarian cancer by 3 separate companies, all having mirror indications. And I think it's exciting because it changes, kind of open up a previous failure, but they're kind of letting the post-market and the physicians dictate which is the most effective and let that be the winner as far as what wins out is whatever works best for the patients. And I can get on that bus. Another is a negative study. I was working with another cancer stem cell inhibitor along a pathway that, again, had preclinical data. Small biotech company had a negative study in another tumor site that they were looking at. I thought it was the wrong tumor site. I thought the ovarian was a much better fit for this agent, but nonetheless, because of all the resources they put into this one, study that was a negative study, they cut the program and then shortly thereafter closed. So those are life lessons I think learned from knowing the landscape. And now, could I have really controlled any of those things? Would I, going back, done anything differently? I'm not sure. I thought they were all very promising ideas. All had some bites on the end of the hook. But I think we just show some of the failures on how to change that to successes. It's just, understanding that we have to adjust to rapid changes rapidly. Got to control what we can control. Relationships are key. Jason hit on understanding what fits the actual idea of the pharmacy sponsor, as well as their business model as well. Understanding that landscape is paramount to having success, specifically with sponsored industry sponsored projects. Another type of I guess it's common sense approach, but under promise and over deliver is a philosophy I try to stick with, and you tend to end up having more repeat customers, and we're able to do that. If you tell them you're going to have 5 people enrolled in 6 months and you enroll 10, you tend to get those people to come back. Even if you knew all along 10 was an easy target. So one of the, I guess, successes, and again, knowing the landscape, there's a company, Gradalis, that I've been working with for a few years now, and they've got some interesting technology in regards to personalized medicine and tumor-directed vaccinations. And so similar to those who might be familiar with TCAR, essentially we're harvesting ovarian cancer tumor at the time of initial surgery. And the company's creating a dual-targeted vaccine to the antigen specific to the tumor cell. So in my opinion, you cannot get more personalized than this than creating a vaccine that only works in a patient that you harvest tumor from, because it's specific to those antigens that are present. It's unique methodology in the sense that it's dual targeted, where there's a stem cell factor enhancement to enhance that antigen presentation to tumor cells, and then has a short hairpin RNA for furin, which is a pathway that helps inhibit the natural immunosuppression of tumors with TGF-beta. And so this company was originally working in sarcoma, and through a mutual friend and some colleagues, had some discussions about how I thought this might fit well within ovarian cancer and the rationale there. And so I think kind of knowing that landscape, having some similar, preclinical data along, the immunogenicity world, we looked at this. And so in a phase 1 study in recurrent ovarian cancer patients, saw that it was well tolerated, with really no dose-limiting toxicities, had promising efficacy in a recurrent setting of an overall response rate of about 40%, and showed some, reasonable survivals that were, promising given the prior lines of treatment. This was a heavily pretreated population, and but the response did correlate somewhat with the lines of therapy. One thing, I guess, is a spinoff on this project, and some of our translational endpoints was an ELISpot test as a predictive biomarker. That is a crude, very generalized marker for immunogenicity, and you're essentially just looking at T cell activation by defined as a quantitative measure of gamma interferon. So it gives you kind of a global picture, but that ELISpot on the tumor cells, to the vaccine in a dish, was able to predict patients that would respond and correspond also to the lines of prior therapy. And so those patients who had 5 prior lines or greater had a negative ELISpot test 100%, which makes sense because these patients have been, bone marrow have been beat on, and their own immune response to tumor was so low, they just really didn't have a robust response. However, if you take those patients kind of, lines 2 through 4, there was a nice split where some of the patients had immune response, some did not. They were ELISpot positive, negative. It's a crude, biomarker, but something we thought was promising moving forward. So since that, phase 1 study, we've completed 2 other ovarian cancer trials. One is a maintenance phase. One is in combination with a PD-L1 inhibitor. And then we have plans for 2 future studies. Really hone in the lab with some specific biomarkers that are, I guess, less crude and more specific. Briefly, another, I guess, success, although this trial hasn't started yet, is one we've been working with through the shared network. Franny has been outstanding working with us. This started out with cabozantinib, which is an oral multi-TKI. A lot of the markers really had a strong rationale in cervical cancer, although the drug has been used mostly in other tumors, including renal cell. We had some preclinical data that supported its use. Originally, I pitched Cabo plus pembrolizumab, and again, I did that knowing the landscape, knowing there were several places looking at PD-1 inhibitors. Could we combine this type of receptor and improve it? The company declined that and wanted a monotherapy, which wasn't unreasonable. There was a back and forth, I guess, intellectual discussion. And someone making a toddy over there? Pass the bourbon, please. Nonetheless, they wanted monotherapy, but soon thereafter, we worked on it, and Franny helped me out a lot on the protocol and getting things lined up with other sites. Pembro received an accelerated approval from the FDA for second-line in cervical cancer. So we then shifted back to, again, the combinational study and are at the point to submit our budget study itself. So again, I'm counting that and chalking that up as a success. Took me a little longer, but given some of the changes. So just in kind of closing, how to optimize successes, minimize failures. Helpful hint: these are a lot of the same things that Jason mentioned. Some of this I mentioned as well as the relationships of both pharma and colleagues. Setting up quarterly meetings. Some of the things that we started doing here is actually setting up a quarterly exhibit fair almost for medical science liaisons, industry sponsors to come in and meet with all our physician scientists talk about cancer-specific research, and we've seen what I think great benefit from that. You have to take charge. I don't care where you are, but especially if in a small institution, you've got to heat the streets and get after it to get these people to come in. It's University of South Alabama and Mitchell Cancer Institute not on the radar like it used to be, like other comprehensive. So it's so important. I hired an administrative assistant who's a nurse administrator to serve as kind of the ultimate liaison. I think a lot of us who are clinical trialists are busy in clinic or seeing patients, writing protocols or in the operating room, and she's been an outstanding constant force for people popping in and medical science liaisons. She's a single contact and has done a great job, and I think it's very helpful in being there when I can't in regards to help running these programs. And we all know it takes trust, it takes time, elbow grease. You got to go to the meetings for your respective field. ASCO, which is the largest oncology association. One of my buddy who's a rep for Bristol Myers Squibb says ASCO is like the prom where everyone thinks they're king and queen. It's an interesting meeting, but those relationships that we build there. Other things that I think has benefited me somewhat, I have on occasion done speaker bureaus, but more importantly, some ad boards for biotech companies looking at pipeline agents and rationale for which disease sites. It's very intellectually challenging and rewarding, and it gets you on the front end of some of these studies and being in charge of where they go and how the trials are designed. I mentioned a little bit the next thing, pitch all the areas in the field. We had a concept for a dual agent with a PARP inhibitor. I didn't just go to, quote, my favorite PARP company or, one of the three companies. I went to all three and said, hey, look, I've got this idea. We've got preclinical data. I'm talking to everybody else. If you guys want to play, let's jump in the boat. That might shoot me in the foot, but I figured that, and then another thing, control what you can control. Some of the things have been there for a while. I think being nimble in South Alabama has really been helpful. We were able to get our IRB turnaround down to about 6 weeks from protocol submission to protocol acceptance, and working with grants and contracts in parallel has been helpful as opposed to doing it sequentially like a lot of other institutions do. Lastly, good ideas are key. Science wins out. Some argue that all trials are compromises either with other colleagues or with industry sponsors. Again, fit the goals of the company, the business plan of the company, but be open to suggestions. I wouldn't compromise the science or what you feel would help patients. I think that is something we all have to dig our heels in is the ultimate goal as we're trying to improve lives. And as I mentioned from some of my failures, expect change. It will happen, but we just all got to keep chopping wood. And at one time I was giving another talk about what is clinical trials and being a clinical trialist most like. And so actually it's like baseball. Baseball is the only sport I can think of where you can have success to plate 30% of the time. And if you do that for your career, you bat 300 and you might end up in the Hall of Fame. And so I think going into it, you got to be able to handle failure, not let it get to you personally, and understand that it takes a lot of attempts to really get one right. But for those that you get right, can be very rewarding. And that's like Forrest Gump. That's all I really have to say about that. And Jason and I'd be happy to take any questions or comments or editorials. Thank you.
Yeah, I was going to say thank you very much, Rod, and also I failed to thank Franny Horn at the beginning. Franny helped pull a lot of these materials together, so thank you, Franny, very much for your help with that. But we're happy to have questions, discussion, comments, whatever.
Before questions, can we thank you both?
Oh, yeah. I'm going to give the hard ones to Rod, by the way. So hard questions go to you, Rod.
So
I'm sorry, I can't hear you. What'd you say?
Yeah.
In my experience, one of the challenging things has been the second discussion with the potential sponsor. So, as a junior investigator, not knowing many of the MSLs, we're typically relegated to submitting ideas through the industry portals. And generally, you'll submit your idea through a portal, and if it's good, they'll contact you for a personal discussion. And I've had instances where things that are good studies or what I think are good ideas sort of fell apart, probably because of the way that I handled that discussion. Simply, it's just not being prepared for what that initial face-to-face or telephone discussion would entail. Does the CCTS have any guidance or resources for junior investigators? Because that's a lot different than writing a grant. And you sort of have to sell your idea, and you have to put on a persona of, this is your expertise. And I guess that's something we're not trained for in science. That's not something we're trained for in medical school. Yeah. Where do we go to learn more about how to navigate that relationship?
That's a great point, and I hate to date myself, but it's been a few years. I certainly we'd be willing to help you through that process individually. I don't know if my colleagues Rod or Mansour or others, if you've got comments about helping junior investigators sort of get through that phase where they may not be as known yet, recognize they've got a good study, and they sort of have to put on a salesman's cap, sort of justify.
Yeah, I got a couple of things. I agree that is sometimes one of the hardest things, especially a junior investigator. I think knowing what the company might want is a good starting point. I try to handle those discussions then and now with like, hey, this is my idea. This is why I think this needs to be this designed this way or in this indication. However, what's the company's interest? Do you guys have a high interest in this other tumor type or this other indication that might also overlap to what I do? Or, what is the thoughts of how you guys would like to see it? And a lot of times you can find a win-win, as Jason mentioned earlier. That helps some, but it doesn't completely give you all the tools that you need to see it, to the finish line. It's a great question and no easy answer, I'm afraid.
Jason, I think it also goes back to mentorship. If you have an idea that's yours, but your mentor proposes it to Merck, Sanofi, likelihood that it's going to go up 2 letters much faster. You can still get to write it, and it is yours. We have this program where we go to ASCO and AACR together as a team, meet pharma. The language and the terminology being used, very important. And at that time, exactly who's making decisions, and you've already been seen by somebody. You were there. I think those kinds of things are very helpful, I think.
Yeah, those are great points, great points. Did you put your finger up?
I did, and
I'll take it halfway down because Mansour basically made the critical point. I think when you're working with colleagues and within that group, there are people at different stages. The most effective group is leveraging the energies and the ideas of everyone within the group. And, if the person with the more contacts can open the door initially to company X, you work together to open the door and you go through it. I think probably if you're working really as a solo person, it's probably a bit harder. One of the advantages I would suggest of working within the CCTS, both at the hub and across the partner network, part of the goal of SHARE is to provide that community to help open the door, that door of access to sponsors, whether it be NIH sponsors or industry sponsors.
All great points, yeah.
And one other thing to add to that, if I may, is that that's a great point. And Jason mentioned a little bit earlier, is that really figure out what your expertise is and what differentiates you from other potential investigators. One thing that we have always used down here at South Alabama is our minority participation on clinical trials, where we have between 30 and 40% of patients on therapeutic trials that are minority African American. And I point out to them that average studies, have 2% or less. And so if they want a real representative population, you should choose us for this type of trial because of that. So I think, having a salesman pitch to it a little bit, knowing what you can really succeed and deliver on can also help.
Indeed, I think leveraging sort of ethnic diversity has been helpful for us here at UAB as well. That's a great point. Other thoughts or comments or?
It's maybe more.
It should be, yeah, just hold
Okay,
it close
for
to you.
people online, not for us.
Oh, okay. I believe in Germany, I left 15 years ago, but as far as I recall, there was a way that the biotech companies were actually coming to universities, to the clinicians, to the, med professionals. So in this way, I believe, now I'm in research here at UAB, I believe we can maybe adopt more that concept of actually having industry financed research and get more biotech companies to finance the research, because that what I see that it could look where your research is having already a different direction, which way you actually proceed, versus if you just do research in the lab or in the clinic on your own, just because your own interest is one direction, versus if you have a very industry-oriented type of research. So maybe we can adopt that and bring the big pharma to finance us, and then the whole field will look different.
Absolutely, you raise a really good point and we as an institution I think are very interested in developing, I'll call them more collaborative, overarching partnership agreements with some key sponsors. In fact, we just had a strategic meeting yesterday where we identified 4 that are a potential for us given our strengths overall as a university. And so those could be very beneficial and fruitful. They can improve efficiencies through the use of master agreements, master contracting agreements. They get to know us and are under our IRB and what we expect. And so those can be very, very beneficial relationships.
Right, that makes sense. And I think what that will actually do help the clinicians or young investigators in a way in clinic or labs to get the pressure of providing grants, needing to publish, that stress of results. If you actually have finances from other sources than from the NIH.
Absolutely.
So.
And Jason, I think one of the things we propose often is IITs are phase 2 small studies. We need to have a repertoire of all our resources, be it molecular imaging, next-gen sequencing, all our tumors are sequenced, we have nanostring, just a repertoire of all those included. When a young investigator goes and talks to somebody, we say whether we have all these elements available here, that might be really a selling point. Whereby, yeah, few patients, but you may get a molecular marker because of all the resources you have. It'd be useful for the CCTS to really compile those. The Cancer Center has some, etc. But we as an institution, and that repertoire can be taken by Andrew or anybody else when they talk to pharma.
That's a great point, and we are in the process of sort of aligning those, and we want to make sure that OSP is labeling the same way that IRB is, that Encore is, but we're sort of pulling those data together now, so we can pull together a resource and say, this is really a sweet spot for us. Or have an external group like Work say, you know what, these are really high sponsors and you don't do very much activity in this space. You ought to be looking at these people over here, given Work's perspective broadly.
Like a brochure or something that you would hand them.
Even an e-brochure, so
Right.
the resources, even most of us could not know what.
Because they have dossiers generally, and we generally don't have something to hand that.
Any other comments or questions, or am I standing between you and a beer? All right. Well, again, thank you all for coming, and thank